In this video, we will show how to upload your own data in multiSLIDE. For this, we begin at multiSLIDE's homepage. Here, we will click Create, and in the form below, we will give the analysis a name, say Analysis 1, and then click the Create button next to the text box. This takes us to the data upload page. Here, the first field is for specifying the species. Currently, multiSLIDE has pathways, GoTerms, and network annotations for human and mouse data. The example data we will use is from humans. The second group of inputs is for uploading a sample information file, which is mandatory. And the third group of inputs can be used to upload multiple data files, one at a time. We will see in a while how these are done. But first, to understand the structure of the data, let's take a look at the input files. Input files should contain the matrix of expression values in delimited text format. Each high-throughput assay or omics type must be in a separate file. Here is an example mRNA data file comprising of 73 TCGA breast cancer tumors. This is a tab delimited data file with rows representing genes and columns representing samples. Each row gives the quantitative expression values of a gene across all 73 samples. Additionally, the first column here contains gene symbols. Such non-numeric columns are referred to as metadata columns. Also, the first row, referred to as column header, specifies sample IDs. For an input file to be acceptable in multiSLIDE, it must follow a few rules. Here, we summarize these rules. First, each high-throughput assay or omics type must be in a separate file. Second, each file should be a delimited text file with one row per gene and one column per sample. Third, it must have a row header containing the sample IDs. Fourth, each input data file must have at least one metadata column containing a standard gene identifier. As we will show later, multiSLIDE accepts a number of standard gene identifiers. A dataset can have multiple metadata columns. Fifth, data files should not contain single or double quotes. And sixth, missing expression values should be either empty or denoted NA. We have a second input data file for protein expression, which has a similar structure. In addition to the data files, multiSLIDE also requires the sample information file. This file specifies which samples are to be read from the input files. This file also lists the phenotypes associated with the samples. For the breast cancer data, we have only one phenotype column that lists PAM50 molecular subtypes. The sample information file also must follow certain rules. First, it should be a delimited text file where each row contains the information for one sample. Second, the first column should list sample IDs with each following column listing one phenotype. And third, the file must have a single row of column headers. Sample information file can also have missing phenotype values. However, these values should be either empty or denoted NA. For each sample ID listed in this file, multiSLIDE searches for a matching column in the data files. Therefore, the sample IDs must match across the two input files and this file. In case a sample is missing from a particular input data file, its expressions will be visualized as missing values. For perspective, here is another example sample information file for a different dataset. This sample information file is tab delimited and has multiple phenotype columns such as age and gender. All these files are available in the multiSLIDE demo data folder in multiSLIDE's GitHub repository. Now, let's go back and upload these files. We will first upload the sample information file. To do this, simply select the file and then specify the delimiter. Notice that multiSLIDE accepts one of these 5 delimiters. Then click Upload. When the process completes, a message reporting the success or failure of the upload will appear next to the white box. We can now start uploading the input data files. To do this, select the type of expression data. We will first upload the mRNA data, so we select that. Then we select the file and specify the delimiter. To upload the file, click the Submit button. Once again, the success or failure will be reported next to the white box. The upload can take a few seconds depending on the file size and the connection speed. On success, the table on the right will show an entry for the data file. Let's call this the files table. The files table lists all the data files that are uploaded and displays the information provided for each file, such as the file name, expression type, and delimiter. To ensure that the correct data has been uploaded and that it has been correctly parsed by multiSLIDE, you can get a preview of the data by clicking the preview icon. To close the preview, click the icon again. If an incorrect file has been uploaded, it can be deleted using the delete icon. Now we can upload the next input file for protein data. This can be done by repeating these steps again. Notice that a second entry for the protein data has been added in the files table. In this example, we only have 2 input files. However, multiSLIDE has no restrictions on the number of data files that can be loaded. Also, there can be multiple files of the same data type. That is, one can upload multiple protein expression data files if required. Recall that each input data file can have multiple metadata columns containing non-numeric data. But at least one of these metadata columns must contain standard gene identifiers. For multiSLIDE to parse the data, we need to specify which column among the metadata columns contain gene identifiers and also what type of gene identifier is present. To specify this information, look at the metadata column information in the files table. By default, we see that the entries for metadata columns and metadata mappings are empty. Click the pencil icon to open a panel where this information can be provided. This needs to be done individually for each input data file. In this panel, the first drop-down lists all the columns present in the file. Each column is identified by the column header. In our dataset, the column gene sim contains gene symbols. So we will select this column and click Add. Here, we need to identify all metadata columns present in the file. So if the file has multiple such columns, select each one and click Add. To delete a selection, click the minus button here. The next drop-down is for specifying which of the selected metadata columns contain standard gene identifiers. This drop-down therefore lists all the columns that were specified as metadata columns using the previous drop-down. Select the appropriate column. Next, select the gene identifier type that this column contains. Note the 8 standard gene identifiers that multiSLIDE accepts. click Add to add the mapping. If the dataset has multiple gene identifiers, you can specify multiple mappings. This is useful if different gene identifiers are available for different sets of genes. Internally, multiSLIDE will map all genes to Entrez IDs. Once all the mappings have been added, click Save Changes. Notice that the information we entered has been updated in Files table. Now, repeat the same process for the protein data. Once the metadata information for all datasets has been added, we can click the Create button to create our analysis. This takes us to the visualization page, where we can select the data to visualize using the selection panel. Notice that the datasets and phenotypes are listed in the drop-downs, indicating that the data has been correctly loaded. We will demonstrate how to select data using the selection panel and create visualizations in the next video.